I'm going to be talking about the neural substrates for flexible and automatic motor sequence execution. The brain exhibits a remarkable ability to flexibly adapt motor behavior to new task demands. At the same time, repeated practice of a task can improve motor performance and render its execution fluid and automatic. In this work, we investigate how the neural circuits underlying automatic motor sequences differ from those underlying flexible motor behavior. All the experimental work I'm going to talk about was done by Kevin Mizes and Ben Silveski at Harvard, and I am working with Sean Escola at Columbia on the modeling side. So in the experimental design, rats were rewarded for pressing levers on a 3-key piano in a prescribed sequence. Rats were trained to execute sequences of 3 lever presses in 2 different kinds of sessions. In flexible or cued sessions, visual cues indicated the order of the lever presses to be performed, and the order varied from trial to trial. In automatic sessions, rats were trained to repeat a single uncued sequence over and over. This overtrained automatic sequence was kept consistent for each rat. To assess the causal contribution of cortical and striatal regions to these flexible and automatic execution modes, our collaborators performed lesions in trained animals. They found that lesions to dorsolateral striatum disrupted automatic but not flexible task performance. In contrast, motor cortex lesions disrupted motor sequence execution for both flexible and automatic execution modes, suggesting a major role for motor cortex not only in sequencing flexible movements, but also automatic ones. This finding differs from prior work suggesting that in motor sequence execution in rodents becomes independent of motor cortex with practice. Interestingly, in providing a potential resolution to this discrepancy, our collaborators found that when rats were only trained on the automatic task, rather than being trained on both automatic and flexible execution modes, their performance was now resilient to motor cortex lesion. This result suggests that performance of similar movements in the flexible context interferes with subcortical consolidation of overtrained motor sequences. What explains these different roles of motor cortex and dorsolateral striatum in these 2 different task modes, and why do the results differ depending on the animal's training protocol? To address these questions, we developed a computational model of the task involving 2 parallel recurrent neural networks corresponding to a motor cortex-mediated pathway and a subcortical pathway encompassing dorsolateral striatum. Both pathways received motor efferents, a recurrent input, and an indicator of context, flexible or automatic, but only the motor cortical pathway received direct cue input from sensory cortical regions. Each pathway outputs force commands that drive limb acceleration, but the actual motor output is computed as a weighted sum of the contributions from each pathway. Inspired by prior work suggesting that cortex tutors subcortical circuits during learning, we modeled the relative contribution of the subcortical pathway as depending on its success in predicting or mimicking the output from motor cortex. A good prediction indicates that subcortical consolidation has been successful and that motor cortex outputs can be ignored. Conversely, a poor copy points to the continued necessity of motor cortex due to its privileged access to sensory inputs. We trained the entire system to perform both the flexible and automatic tasks and found that the key lesion results in the experimental data were explained by the model. DLS lesion disrupted automatic but not flexible task execution, and MC pathway lesion disrupted both, but when the model was trained only on the automatic task, execution was once again robust to MC lesion, as in the experimental data. This suggests that the lesion results described experimentally can emerge naturally from a task optimization in this 2-pathway model of motor learning. In particular, in the model, this phenomenon arises because the subcortical pathway lacks the sensory inputs needed to predict motor cortical outputs. Because of overlapping representations employed by DLS during the flexible and automatic tasks, this unpredictability interferes with the consolidation process even for the automatic task mode, where the sequence is in principle fully executable by the subcortical pathway. One additional prediction of our model is that both motor cortex and DLS should represent information about egocentric movement kinematics, but that lever sequence information should primarily be represented in motor cortex. This prediction agrees well with experimental data, where a substantially higher fraction of recorded motor cortex units are selective for the identity of the upcoming lever press. So in conclusion, we've provided evidence that distinct neural pathways, cortical and subcortical, are involved in flexible and automatic motor sequence execution in rodents, and that this distinction can arise in a two-pathway neural network model in which a cortical pathway has privileged access to sensory input relevant to the task, and in which continued cortical engagement is driven by subcortical prediction errors. Thank you.